Alright, welcome to video 2 of this LD50 lab. So in this video I wanted to go over some tips for designing your experiment and some things that you should think about. This is also in the instructions here online. And pro tip, if this is too small, you can click over here and zoom it in and boom, you got it much bigger. Okay, so over here I've put in the recommended schedule. It takes a few days for potentially any seeds to germinate, so you want to set this up today, hopefully on Tuesday, wait a few days, and then collect your data. In the days that you're not collecting the data, you might want to take pictures and stuff like that just to write down some observations. Great, so like I said in the previous video, you're going to be designing your own experiment. You're going to basically get to decide how many mung beans you want to test, the salt concentration, and the time that you decide to let the petri dishes sit. So important things to remember when you're designing your experiment. First of all, water evaporates, yeah? And if we're creating a salt solution, we know that the water will evaporate, but the salt will stay in. So what that means is we want to be really careful of any evaporation because that's going to change the concentration of our salt solution. So make sure that when you put in the paper towels into your petri dishes, you don't have anything hanging out because that's gonna leave for evaporation. You might also try to consider sealing your petri dishes around the edges here or completely covering all of them with something just to prevent the evaporation from happening. Right? Next thing, you are given kind of a limited number of mung beans, right? Unless you have some at home. You need to decide how many mung beans you want to put in each of your petri dishes. And of course, you don't want so few that you don't get good data, right? Not statistically significant data. But you also want to make sure you have enough mung beans for all of your petri dishes so that all of them have the same amount and that they're not overcrowding in the petri dish. So none of the mung beans should be touching. There should be a little bit of space in between each. Cool? Next thing to think about here is in your lab kit, I gave you these little packets. These are packets of iodized salt and they're about 0.75 grams each. So I think I gave you around 7 packets or so, so you should have about 5 grams, right? 5.25 grams total of salt if you were to use all of the packets. So keep that in mind. That is the salt that you have for this experiment, so don't use it all or use it and then realize you need more of it, right? Unless you have salt at home and some sort of weighing or measuring device for the salt. And then the next thing is we want this test to be reasonably accurate and representative. And the ocean is only 3.5% salt, right? The solution of the ocean. So when we make our solutions, we don't really want to go over 5%. It's not really realistic into what farmers may be facing or anything like that. Great. And then the next few things here is make sure you are accurate with all of your measurements, right? So each of these is 0.75. Don't round it up to 1 gram, 0.75 grams. And you have your graduated cylinder to measure your water. You also probably have one of these which also has measurements of milliliters along the side. So please don't eyeball your measurements for your water, even though that's what I'm going to be doing most of the time because I don't have a measuring thing except for this. Okay, and then finally, don't forget, in all good experiments you have some sort of control. So make sure you set up a control with no salt. So that's just going to be plain water on there. Okay, so let's walk through our design. First of all, choose how many mung beans you're going to have in each petri dish. It could be 10, it could be 15. Depends on how many petri dishes you're setting up. Also depends on how many mung beans you have, right? Decide what concentrations of salt solutions you want. So remember, you need a control, which is going to be 0% salt. Your highest one should be no higher than 5%. But then you get to decide what goes in the middle. Do you want to do 1%, 2%, 3%? Do you want to do 1.5, 3.5, so on? Or do you want to do 0, 0.5, 1, 1.5, 2, and then a really big jump. Alright, so think about what solutions you have. You have 5 petri dishes, and one of them has to be the control, and then you get to decide what these other 4 are. Cool? Once you have decided that, you're going to do your calculations to figure out, well, how am I actually going to make that with water and with salt? Cool, and so here are the formulas for the calculation, and I'm going to do this in a separate video, so check the playlist for the separate video on how to do all of these calculations here. And then finally, last bits of your designing phase is decide how many days you want to run the experiment. I recommend between 2 and 5. I think 3 is probably the sweet spot number. And then decide how you're going to stop the evaporation from happening. You might decide to take some tape and wrap it all the way around the petri dish, or you might decide to wrap it in foil or something like that. Cool, so those are the things that you need to think about for your design. And then as you are doing that, feel free to open up the LD50 lab wrap-up, and you can start answering some of these questions here, the pre and the during the lab questions. So thinking about what question are we trying to answer, what is independent, dependent variable, what is your hypothesis, that sort of thing. So this document here is what you're actually going to be turning in. So make sure you put all of your stuff, your answers, your data, everything into this document here, the one that's called Lab Wrap-Up. This one is just the instructions. You won't be able to write on this one. Okay, cool. So I'm going to take you through the calculations in the next video. Otherwise, good luck designing.